Hi guys, um, here I have a Cambridge International AS and A-level exam paper. It's a biology exam paper. It is paper 1. You know that paper 1 is multiple choice. This paper was written in June 2025. So in this video, we are going to work out the solutions for questions 1 to 10. Let's get started. All right. So here I've got question 1. So question 1 says, you can see that it reads, plant cells are stained and then viewed with a simple light microscope using daylight as the only light source. Which cell structure are clearly visible at a magnification of 400 times? So, chloroplast grana, no, it's not visible at this magnification. Lysosome, no, and the answer is actually nucleoli, right? Nucleus and nucleoli are visible under this magnification. The ribosome is too small, can't be seen with the light microscope. Okay, let's move on. One was a piece of cake. Number 2, the diagram shows an image of a cell with a scale bar. The scale bar Z represents an actual size of 15 micrometers. Distance Y represents the diameter of the cell image. So this is what we see here. Okay, it's a cell, right? Of course, this looks like a red blood cell, right? Y is here represented, is the image size, and this is the scale bar, which is 15 micrometers real size. So what you have to know first is magnification. Magnification is given by image size over real size. Real size is actual size. You have to know that, right? You also have to know that if you want to get real size, okay, because they want us to get which calculation, the question here is which calculation to find the actual diameter of the cell is correct. You want to know the formula here, but the standard formula here is real size is equal to image size over magnification. Real size is actual size. So if you already know this formula, real size, image size over magnification, you have to just proceed. So it means that image size is actually equal to, so just get back to the scale bar. So from the scale bar, you can actually get the magnification. How do you get the magnification for the scale bar? From the scale bar, which is given here as Z, magnification is going to be size of scale bar. So you need to use a ruler so you can get the size of scale bar, right? Represented it like this, SCB over real size of scale bar, so actual size of scale bar. So you take this and you now replace it in this formula here. Okay, you substitute it in here, you're going to get image size over size of scale bar. Actually this is supposed to be S, not C, right? S, not C. Okay, size of scale bar, right? So image size over size of scale bar over real size. Just use mathematical rules here, and you are going to have image size real size, right? We're going to have real size is equal to image size times, okay, this was the denominator. Now I want to make it, we're dividing is over SSB over RS, so just use that mathematical rule. Now this is actually a division. We want to make sure that this is a multiplication, so we're going to invert this equation here, or in this case this fraction here, you invert it. All right, you want to invert it, so it's gonna be image size times, so now by inverting, so it's gonna be times real size of the scale bar, which is this one here, okay, real size of the scale bar is now here, over size of the scale bar. So now it gives you this formula here, and this formula here is actual size is image size times times real size of the scale bar. Okay, they told us that the real size, the actual size of the scale bar is 15 micrometers over size of the scale bar. So this corresponds to this option here, option C. Right, it corresponds to option C. So this is the correct answer for number 2. Okay, let's move on. Let's see what we can do. In number 3, right? Let's see what you can do in number 3. So number 3 here says the diagram shows a stage micrometer with divisions of 0.1 millimeters viewed using an eyepiece reticule. So that's what we have here. So the eyepiece reticule is here, okay, a label here. Okay, this is the eyepiece reticule. This is the eyepiece reticule. Right, there we go. From 0 to 100 is the eyepiece reticule, and we have the stage micrometer here. This is the stage micrometer, right? So it says the diagram shows a stage micrometer with divisions of 0.1 millimeters. Here is 0.1 millimeters, then 0.2. So that means that 100 in the piece, in the eyepiece reticule, corresponds to 0.2 millimeters in the stage micrometer. Okay, you have to know that, right? Now, pollen grain were grown in a sugar solution and viewed using the eyepiece reticule. Diagram 1 shows the pollen grains at the start during and then says diagram 2 shows the pollen grains after 2 hours. Okay, after 2 hours. Now you can see the diagram here. This is the diagram, right? Pollen grains are still too small. The pollen tube in this case, too small. Then this diagram here on the right side, you see 1, 2, 3, 4, 5 pollen grains, but you can use this one here. All right, actually make sure you have this so you can measure. All right, you can see the measurement here. You can see that according to the eyepiece reticule, all right, it has grown from 25 to 35. That corresponds to 10 units. That's it. Now we are saying that the 100 in the eyepiece reticule, we need to calibrate it, corresponds to 0.2 millimeters in the stage micrometer. All right, so if we want to know each unit corresponds to what, so we've got 100, so we're going to divide 0.2, okay, this 0.2 by 100, that is actually from the eyepiece reticule, and we get that per unit is going to be 0.002 millimeters. So each eyepiece reticule unit corresponds to 0.002 millimeters. All right, but we want to convert this into micrometer. we know that 1 millimeter corresponds to 1,000 micrometers. So 0,002 times 1,000 is 2 micrometers. And you also know in this case that each eyepiece reticle, according to this convention, corresponds to 2 micrometers. All right. Now we want to know the growth. The question here is, what is the growth rate of the pollen tubes? The growth rate of the pollen tubes. So the growth rate of the pollen tubes is going to be given by growth over time. Growth, all right, or increase over time. So we know from this diagram here on the right that it has grown 10 units according to the eyepiece reticles, and we have already calibrated it, right, according to these calculations here. All right, so growth rate is going to be 2 times 10 units here, which is 20 now. It's here, all right. It is 20 now. Now, how many hours? After 4 hours. So it's going to be 20 over 4, which is actually 5. And that's the answer, right? So the answer is 5. So the answer here is going to be 5 micrometers per hour, right? That's why we had to convert this to micrometers, because the options here are given in micrometers. All right, now we get to number 4. What are found in chloroplast and also in mitochondria? DNA is found in mitochondria and chloroplast, yes. Ribosomes are found in both organelles, that's also true, yes. And mRNA, if ribosomes are there, of course they're going to have their own mRNA. If they've got their own DNA, they must have their own mRNA, and then ribosomes for protein synthesis. So it's going to be 1, 2, and 3, so option A, right? Option A. So I have the side note here. They can produce their own proteins because they have their own DNA and ribosomes from and ribosomes. So from DNA they get their messenger RNA, but most importantly, you have to understand that most proteins actually come from the cytosol, from the cytoplasm. All right. I'm gonna actually make sure that I think that this video is gonna be too long. I wanted to actually work out 10 questions. Let me do it in another video. All right, another video I'm going to proceed from 5 hopefully to 10. All right, so far we are going to stop here. Thank you so much for watching this video.